I'm associate professor in the Department of Cellular and Integrative Physiology at University of Texas Health Science Center, San Antonio. Today, I will have a one-on-one meeting with Dr. Qutuba Karwi, who is coming from the University of Alberta, Edmonton, Canada. He presented his work during the poster session 2A regarding myocardial infarction, ischemia, and oxidative stress and cardio protection. Today, we would like to know more about his work, and I will ask Dr. Qutuba to give us more about the significance and the big findings of his work entitled Branched Chain Keto Acids, Not Branched Chain Amino Acids, Impairs Cardiac Insulin Sensitivity by Disrupting Insulin Signaling in the Mitochondria.
Thank you very much, Dr. Bopassa. I really appreciate this opportunity to talk more about my work that I'm going to present at the BCVS. First of all, I would like to thank the BCVS organizing committee for giving me the opportunity to present my work in this moderated poster session, and also for recognizing my abstract and the Paul Dudley White International Scholar Award, which is a highly prestigious award, and it's a great recognition for this work. So what I'm actually trying to share here in this poster is some new data that we got recently, where we're trying to understand the importance and the role of branched-chain amino acids in inducing on the contribution to cardiac insulin resistance in the failing heart. So we all know that the branched-chain amino acids are important energy substrates and signaling molecules within the heart, and alterations in branched-chain amino acids, or as I will be referring to as BCAAs, metabolism have been linked to the development of cardiac insulin resistance in the failing heart, and in different pathologies like diabetes and obesity. So this is a general consensus in the field of energy metabolism. However, whether it's the accumulation of the BCAAs themselves or their metabolites, namely the branched-chain keto acid or BCKAs I'll be referring to, are the main mediator of the cardiac insulin resistance in these pathologies is not known. Also, how these metabolites or the energy substrates are inducing cardiac insulin resistance in the failing heart is also not known. So we ask two main specific questions in this work, which is one, is it BCKAs that are mediating the insulin resistance in the heart? And whether it's BCAAs or BCKAs are the main mediator, how are they mediating this action? So we wanted to understand the mechanisms. So what we use as an experimental approach to do is to try to raise BCAAs levels in the heart and reduce BCKAs by looking to the insulin signaling as well as the cardiac function in these hearts. And
can I ask you, because it is, I would like to know something, give us the difference between BCAA and BCKA, please.
Yeah, absolutely. So BCAAs are the the amino acids that we get when we eat, for example, certain type of fish. So those inside our body will be metabolized into branched-chain keto acids. So basically, we don't ingest branched-chain keto acids. What we ingest as one of the types of the amino acids, the BCAAs, the branched-chain amino acids, and there are three types: leucine, isoleucine, and valine. So these are the three types of branched-chain amino acids that we are talking about here. And once they are inside our body, they will be metabolized into branched-chain keto acids. So the genetic experimental approach that we use is knocked out and that's responsible for metabolizing the branched-chain amino acids into branched-chain keto acids, specifically in the heart. And what we see is once we knock out this enzyme, we see a significant increase in the branched-chain amino acids level in the myocardium, and a significant decrease in the branched-chain keto acids because there's no enzyme that will convert the branched-chain amino acids into branched-chain keto acids. So, and then we looked what would happen to the insulin sensitivity in the heart in this situation. So when we look at the ability of the heart to oxidize BCAAs, we see that it's impaired because the heart can't oxidize BCAAs anymore because we already knocked out the first enzyme in the BCAA oxidation pathway. When we looked at the insulin sensitivity as an indicative of insulin-stimulated glucose oxidation, what we saw was really interesting. We saw an enhancement in the insulin-stimulated glucose oxidation in the BCAD-M knockout mice, and I should have said that the enzyme that I'm talking about is the branched-chain aminotransferase 2, which is a mitochondrial enzyme that is responsible for converting the BCAAs into BCKAs. So what we see in those BCAD-M knockout heart is that we see an increase in insulin-stimulated glucose oxidation rate, suggesting that cardiac insulin sensitivity is enhanced with the reduction in BCKAs, not BCAAs. What we also saw is an increase in the left ventricle mass, which is an indicator of cardiac hypertrophy in the BCAD-M heart, suggesting that the accumulation of BCAAs is triggering cardiac hypertrophy, and that was associated with increased activity of the mTOR signaling pathway, which is a well-known signaling pathway that is linked to cardiac hypertrophy. So what we wanted to do next is to look into what will happen if we reverse the scenario. If we increase BCAAs and we decrease, BCKAs and decrease BCAAs. So what we did is we randomized two sets of mice hearts to be perfused with vehicle or with high levels of BCKAs, levels that we see in diabetes and in obesity. And we were really intrigued by the results. So what we saw is when we raised myocardial keto acids or BCKAs, we see a significant reduction in cardiac insulin sensitivity we see a decrease in the insulin-stimulated glucose oxidation, as well as we see a significant reduction in the insulin signaling pathway activity. So what we wanted really to understand is how BCKAs are mediating this reduction in cardiac insulin sensitivity. So what we see in the BCADM knockout heart is an increase in the activity of a kinase called protein kinase B, or AKT. So this is a major component of the insulin signaling pathway, and when it's increased, that is an indicative of an increase in the insulin signaling pathway activity. So what we see when we knock out the BCADM enzyme, we see an increase in the AKT activity, suggesting that a reduction in the BCKAs levels in the heart will enhance the activity of the insulin signaling pathway. But when we looked at the downstream activator of AKT, kinases like GSK3 beta or glycogen synthase kinase 3 beta, we see that its activity is not changed. So what we asked next is why only AKT is changing and not its downstream activator. AKT work by other investigators showed that AKT can be translocated into the mitochondria following insulin stimulation, and this translocation was associated with enhanced cardiac glucose oxidation rates. So we really wanted to understand whether this mechanism plays a role in the BCKA-induced cardiac insulin resistance. So we isolated mitochondrial and cytosolic fractions from BCKA, high BCKAs treated hearts, and we looked at the translocation of AKT within the cytosol and the mitochondria. And what we really observed was really interesting. What we saw is when we hit the heart with insulin, we see increased mitochondrial translocation of AKT, and this increased translocation of AKT to the mitochondria was associated with enhanced activity of pyruvate dehydrogenase, which is the rate-limiting enzyme of glucose oxidation inside the mitochondria. However, when we perfuse the heart with high level of BCKAs, we see that this translocation was completely blunted, and this was also associated with a decrease in the activity of PDH, suggesting that indeed BCKAs are impairing cardiac insulin sensitivity, an effect which was associated with a decrease in the mitochondrial translocation of AKT to the mitochondria. So what we think is happening is BCKAs are inducing cardiac insulin resistance by impairing mitochondrial translocation of AKT to the mitochondria. And it's BCKAs, not BCAAs, are responsible for impairing mitochondrial insulin signaling in the heart, while BCAAs are playing an important role in triggering the hypertrophic signal in the heart. It's not BCAAs. Sorry.
Because you are trying to point the key role of AKT signaling in the mechanism. Have you tried a pathway like that? Because we know that phosphorylation of AKT is coming from PI3K, Have you tried to use the inhibitor to block this and see whether your mechanism, your pathway is abolished?
If I understand your question correctly, Dr. Bopassa, you're asking whether we tried to use a PI3 kinase inhibitor.
Yeah.
Okay, so the answer is no, we haven't tried this yet. For one reason, which is we think that what we know from the literature, that inhibition of PI3 kinase will inhibit the phosphorylation of AKT, which is an effect that we see when we perfuse the heart with high level of BCKAs.
Yeah.
So in other studies that we are doing right now, we actually see that when we inhibit PI3 kinase, we see a significant reduction in mitochondrial translocation of AKT. So I think the phosphorylation of AKT in the cytosol is what triggered this translocation into the mitochondria.
There will be, I believe, if the room was full of people here, someone should ask you this question.
That's right.
PAKT is really going everywhere. It is like highway. How sure are you that in this condition, your mechanism is only impacting mitochondria? To why it is possible that activation of AKT can activate also something else?
That's a very important point. Actually, you are absolutely right, Dr. Bopassa. The effect of AKT on triggering the activity of PDH inside the mitochondria doesn't mean that there isn't any other pathway that may be involving in modifying glucose oxidation in mitochondria. However, we know that PDH is the rate-limiting enzyme in modulating glucose oxidation inside the mitochondria. And what we see, as I mentioned before, is when we inhibit this mitochondrial translocation of AKT to the mitochondria, we actually seeing a significant inhibition of PDH, suggesting that at least in part, mitochondrial translocation of AKT plays a prerequisite role in triggering the activity of PDH, and as a result, triggering or stimulating cardiac glucose oxidation rate.
One more question. I think we have some time, and it is very. I like your work, and it is some enthusiasm. These results, because you use the heart, could we use the same? Could we see the same thing in the brain of a kidney? Is it possible to see?
To answer your question, that's a really important question. So to answer it, I don't have any data to say to support any suggestion right now, but this is work that we are doing right now in different models of obesity and diabetes. For me, my interest is in the heart. I've never done anything with the brain, but it's possible. But if you ask me, do you have any data to support that? I will say no.
Okay, the last question. This is more general because you know someone who is not a scientist can follow you. Say that the BCKAs are coming from the BCAAs, right? What could be your advice for people to eat less BCAA or to eat more in general in terms of nutrition?
That's a really important question. So when it comes to the heart, direct measurements of cardiac branch chain amino acid oxidation rates in our lab showed that BCAAs actually contribute to 1 to 2% of cardiac ATP production. So that's very minimal. Let me give you an example. So when we compare BCAAs to glucose or to fatty acids are the major contributor to cardiac ATP production, with 40 to 60% of cardiac ATP coming from fat. Glucose contribute to 20 to 40% of cardiac ATP production. So these are the major two oxidative substrate in the heart. What we think is happening is changes in the contribution of BCAAs to cardiac ATP productions are not really the main contributor to the alterations that we see in glucose oxidation. We think that the role that BCAAs play in modifying cardiac insulin signaling pathway in the heart is more important than their contribution to as an oxidative substrate to cardiac ATP production. So a really relevant example here is bodybuilders. They take this BCAA supplement to increase their muscle mass or body mass. So this is really very consistent with the data that I just showed you, that it's actually not BCKAs that are triggering hypertrophy, but it's actually the accumulation of BCAAs that we see when we knock out BCADM in the heart. We see it in the heart when we increase BCAAs level in the heart. We see an increase in the left ventricle mass, similar to what happens in the muscle. So that is actually supporting our findings that the accumulation of BCAAs trigger hypertrophy. The accumulation of BCKAs is cardiac insulin resistance. So I'll definitely say that reducing the amount of BCAAs in your food could be beneficial if you are obese, if you're having any status of insulin resistance.
Thank you for coming to present your work. I believe people who will follow this video will really like. I enjoyed to hear your explanation. Thank you so much.
Thank you for having me. It's a great opportunity to speak to the AHA Science News. It's great. Thank you so much.